Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 205 for the podcast. Today, I'm delighted to welcome Andy Parker, CEO of Step Pharma. With over 25 years of experience across AstraZeneca, Shire, Zealand Pharma, and venture capital, Andy has led Step Pharma since 2019. The company is pioneering a targeted approach to cancer and blood disorders by inhibiting the enzyme CTPS1. Their lead candidate, dencatistat, blocks this pathway that certain cancer cells and activated immune cells rely on, while sparing healthy cells that use the related CTPS2 enzyme. In this episode, we'll dive into the science behind this mechanism, explore Step Pharma's expanding pipeline, and discuss their recent €38 million Series C financing. We'll also look ahead to the future of precision oncology. I hope you enjoy my conversation with Andy Parker. Andy, welcome to Beyond Biotech.
Dylan, thanks very much. It's a pleasure to be here.
Andy, way back when, tell me, what was it that got you interested in science to begin with?
I think that's easy. That tracks back to school days. That goes back to having a fantastic biology teacher that brought so much enthusiasm to the subject that it just got me hooked, and continued on from sixth form to where I am today. Actually, I regard my career has very much been focused on my passion. So it's been that's the story, really.
Where did you go on to study after sixth form?
I did my degree at the University of Surrey. And actually, even back then, I was really focused on thinking about industry as an opportunity. So I did a degree that offered me a year as a placement in industry. So I went and worked with Zeneca before it was AstraZeneca at the time. And then I went on to do a PhD at the National Institute for Medical Research in London at Mill Hill. Sadly, not there anymore. And then a postdoc, and then came back. I did a postdoc in the US with Johns Hopkins Medical School, and then I came back to Europe and straight into industry and been in industry ever since.
You never had that pull towards academia? You never wanted to move into that side of the business?
I think not. I think I was always more interested in the translational aspects of what I'd been doing. And I sort of moved from microbiology to genetics into cancer biology, and was really, intrigued with where this might go from a translational perspective, and starting to think about drug discovery and then ultimately on into drug development.
You mentioned Zeneca, but you also worked at Zealand Pharma, at Shire. You even worked in venture capital as well. Tell me a little bit about that.
Yes, I went into venture after I spent some time at Shire. And while I was at Shire, I got involved with the investment group that Shire ran, and that spurred me into having some interest there. I also did an MBA part-time in order to learn a little bit more about business models, etc. And I had an opportunity really to join a VC fund in Geneva and to spend a number of years there. But ultimately what I really, really enjoy is being inside a company and building from within. So I think the VC experience was fantastic. It taught me an awful lot, but it also led to my decision to then move on and join Zealand Pharma back in that sort of operational role.
It seems then you've seen biotech from quite a few different perspectives, from the capital side, also within top pharma, some slightly mid-sized firms as well. What sort of perspective has that given you on the industry?
I think the time that I spent with Janssen originally, and then the sort of 10, 12 years that I spent with AstraZeneca, gave me a fantastic training, but also a good understanding of how big pharma operates, what big pharma is looking for. And then I think I had a natural progression into smaller companies, from small startups like Opsona that are rather similar to Step, to Shire, which wasn't a biotech company, but it had a very biotech feel when I was there. So I think I've seen different ways to do science and to do drug discovery and early development, from the big organizations with many functions all within, to the smaller organizations like Step, where we're outsourcing as much as we can and get a good understanding of how to do things in a very cash efficient and time efficient way through all of those experiences.
Now, you joined Step Pharma as CEO in September 2019. What attracted you to the company? What was it about their approach that pulled you in?
I think I was drawn in by a number of different elements. At the time when I was looking for that CEO role, I knew a number of the investors in Step. So there was a good connection there. I was absolutely intrigued by the mechanism. I mean, this is a mechanism that's driven by human genetics, and that human genetic phenotype is so clean with respect to the consequences of the loss of CTP synthase 1 that was just intriguing to me. And then the chemistry program had been run by Signature Discovery in Nottingham, and a lot of people there were ex-AstraZeneca colleagues of mine. So I had a lot of touch points to really get into the science, ask about the chemistry, knew the investors well, and then mechanistically it was just so interesting to be able to work on something where if the pharmacology could recapitulate what you'd seen in genetics, you knew you were going to have a really interesting drug.
We're going to jump into the science of Step Pharma in just a second, but I want to ask you one more thing. It's Step Pharma, the headquarters. It's located in France, but it's very close to Switzerland. You've been based in Geneva, as you said before, and for a while now. You know, we all hear about Basel, we hear about Zurich and the money that's there and the pharma companies that are there. But is there a good ecosystem for biotech in Geneva too?
Yeah, there's actually a great ecosystem. And I think if you put Lausanne into that mix as well, between Geneva and Lausanne, actually, there's an awful lot of biotech activity. In the Geneva area, you've got companies like Novimmune, like Ionctura, Addex. In the Lausanne area, you've got Asimune, Asineuron. There's an ecosystem there with a whole pile of companies. I mean, after Merck, Serono left Geneva, actually then their buildings became Campus Biotech, where they incubate an awful lot of companies. So whilst Basel and Zurich might have the bigger names, and there is actually a really vibrant ecosystem around the Geneva area.
Let's talk a little bit about Step Pharma now. What would you say the core mission of the company is?
I think the core mission is to maximize the benefit that inhibition of CTPS1 can bring to patients. We have something entirely novel in our hands, and I think it's behoving on us to actually now try to really demonstrate how that can be used in multiple indications in order to bring as much benefit as possible.
Tell me a little bit about this enzyme, this CTPS1. Maybe explain in simple terms why certain cancer cells and activated immune cells depend on this particular enzyme.
Yes, so we target a pathway called the pyrimidine synthesis pathway. So this is a pathway that's actually producing CTP, and CTP is an integral component of DNA and RNA. So essentially what we're trying to do is to block DNA synthesis in cells. And if you block DNA synthesis in cancer cells, for instance, then what happens is that they stop dividing. And if you can hold them there for long enough, then what happens is that those cells go into apoptosis, into cell death, and actually then die. So that's a mechanism that's actually quite well understood in the cancer setting. But what's different here is that most approaches that try to block nucleotide synthesis don't do that in a tumor-specific way. And so consequently, if you block nucleotide synthesis throughout the body, you also block cell division in those cell types that are actually important for normal life. And what you usually see in a broad nucleotide synthesis inhibitor is gastrointestinal tox, because you've blocked replenishment of the gut wall, you end up with mucositis, with diarrhea, and that prevents and has prevented development of drugs that target this particular pathway. The reason that CTPS1 is interesting and different partly comes from the human genetics story. So Alain Fischer, one of the founding scientists, he identified a founder population of kids back in 2014, they were in the UK, who were unable to mount a response to a viral infection. And what they showed was that inability to mount that response was due to a mutation in the gene that codes for CTP synthase 1. So essentially a null, so almost a complete loss of function, meant that your lymphocytes couldn't proliferate. And then what was most important was that CTPS1 also has an orthologous enzyme, CTPS2. And every other tissue type in your body, in the absence of CTPS1, can use CTPS2 to make whatever CTP they need to carry out normal function. So that was a sort of eureka moment that if we could truly target CTPS1 selectively, we could block aberrant lymphocyte proliferation in, for instance, the lymphoma setting. And that is unique in the pathway that we're targeting. So that pathway has sort of 6 or 7 biochemical steps. And every step in that pathway, there's one enzyme that can catalyze the substrate to product conversion, except that final step where we find ourselves with CTPS1 and CTPS2. So really what we're doing is exploiting the situation where everything is dependent upon CTPS1. So in lymphoma, lymphocytes can only proliferate if they have CTPS1. Doesn't matter that they have a low level of CTPS2. And then we've gone on to find other situations where CTPS1 is an absolute dependency in solid tumours and also then in essential thrombocythemia. So it's really finding that exquisite point in time where you've got that dependency. That means you can block nucleotide synthesis, have the desired effect, whilst sparing all the other tissue types.
So you've got this dependency, you understand the mechanism there, and then we've got dencatistat. Where does this fit in?
So dencatistat is actually the product of some fantastic chemistry from the guys at Signature Discovery in the UK to truly optimize a molecule that is a selective inhibitor of CTPS1, so 1,300-fold selective for CTPS1 over CTPS2, that has absolutely fantastic drug-like properties that have now been borne out in the clinic in terms of fantastic dose proportional increase in exposure, good oral bioavailability, and actually a really clean safety profile. So it is a first-in-class drug targeting CTPS1 and moving forward rapidly in multiple indications.
So the idea here is that we're going to be able to treat cancers, we'll treat tumors, blood disorders by turning off the tumor and stopping its ability to grow while maintaining just the healthy growth of other cells in the body?
Essentially, yes. So in the lymphoma setting, we believe that lymphocytes are dependent upon CTPS1. So inhibiting CTPS1 in lymphoma should specifically target the tumor, sparing all other tissue types that can still use CTPS2. In solid tumors, what we have established, because intuitively solid tumors wouldn't have been a sensible place for us to try to exploit the drug, because we know that cells derived from origins other than a lymphocyte are able to use CTPS2. But what we have established is that there's a very high prevalence of the loss of CTPS2 in cancers. And that can either be a genomic loss, so that a region of the chromosome has been deleted in the cancer, or it can be an epigenetic shutdown of expression of CTPS2. But what that means is that the tumor now is entirely dependent upon CTPS1 to make it CTP. So you can very specifically target that tumor whilst leaving all the other tissue types untouched, essentially. And so that's the basis of a study that we have running, where we try to select patients for that loss. And then in essential thrombocythemia, it's the same situation, where what we know is that human megakaryocytes don't express CTPS2. So they are the only tissue type in the body, actually in normal physiology, that have switched off CTPS2 and have this dependency on CTPS1.
I think what makes this really interesting for me is that you're not developing it for just one indication. You're not developing it just for one type of cancer there. It's really multiple indications. It's what we call that pipeline in a product strategy. Is this a really obvious choice for the company? Did anyone think to go in this direction, or was it really the only choice?
I would say it's always hard to decide at the beginning that you're going to create a pipeline in a product. I think it's driven by the science, and that's very much what we have done. So we started the lymphoma study because human genetics said there's a dependency there. we then established that there was this genetic loss in solid tumors, which created an opportunity. And so then we built the case for how we would advance this into solid tumors, creating an opportunity to go into multiple indications because that prevalence is actually very high. And we focus right now in on ovarian and endometrial and lung cancer. Endometrial, we just recently got an EIC accelerator grant specifically to exploit the opportunity in endometrial cancer and really bring out a women's health focus in the oncology space. And then actually the ET story came very much from what did we learn in lymphoma? We learned that we have an effect on platelets that in the solid tumor setting we have to manage. So we spent a lot of time trying to figure out how do we avoid the effects on platelets. And so in aggressive cancers, we dose the drug 1 week on the drug, 1 week off the drug, so that then you cycle your platelets, but you don't have any clinical sort of manifestations. But ET is actually an indication where you want to reduce platelet count because ET is a leukemia that's driven by overproduction of platelets. And in order to manage that condition, you need to bring those platelets back into the normal range. So we saw an opportunity perhaps to exploit what was a safety signal in solid tumors and lymphoma and turn that into the therapeutic endpoint in ET. But the only reason that we could then start that study was because in lymphoma and solid tumors, we dosed enough patients to convince ourselves that in a chronic indication, we had a really clean safety profile. And we have no recurrent AEs in our lymphoma and solid tumor study at doses that are 5 to 10 times higher than we will be using in the ET study. So we recently kicked that program off. We dosed the first patient right at the end of last year, and we have 9 patients on study and all are responding to the drug. So some really compelling data in what is quite different as an indication. It's a chronic indication diagnosed in your 50s and 60s. First line therapy is effective, but 25% plus of patients become refractory or intolerant to that first line and are looking for a better second line opportunity. So I would say we didn't start at the beginning saying we were going to build a pipeline in a product, but the science taught us and the investors supported us to enable us to broaden out to now have 3 different indications running concurrently.
So you've got 3 different clinical trials, and you've got the relapsed and refractory trial that you just mentioned. You're looking at solid tumours as well, and the third one?
And essential thrombocythemia is the third.
Okay. And at what stage are these trials right now?
So the lymphoma study is in dose escalation, very close to what we predict would be the phase 2 dose. So we will soon be going into the safety expansion cohort. And at that point, we will establish what degree of efficacy we're seeing in a defined patient population. And that'll help us decide towards the end of this year where we're going from a phase 2 perspective. Then the solid tumor study is roughly similar situation in terms of escalation. But what we did in the solid tumor setting was to dose escalate in all comers solid tumor patients without any biomarker selection in order to get us to the phase 2 dose as quickly as possible. And we're pretty much there now. So what we will then do is look at 10 to 20 ovarian cancer patients, endometrial cancer, and lung cancer patients, all biomarker selected. So safety expansion cohorts, which will then determine, is our hypothesis correct? And can we demonstrate that we have activity across multiple cancer types? So we can really understand, is this a pan-cancer therapy based upon the biomarker? And that will also then direct what we do in phase 2. So we'll get to the end of that phase 1b expansion cohort towards the end of next year. And then the ET study, as I say, has 9 patients on study so far, all responding. This is now a phase 2 study, so we've advanced that quickly into phase 2. We aim to recruit 50 patients by summer next year, with the expectation that we can be phase 3 ready in that study by the end of next year. So moving very quickly in ET. Because of the nature of the study, the way that it's designed, we get data very, very fast as opposed to what we're doing in lymphoma and solid tumors where we're sort of beholden to 2-month cycles to get scans on patients. Then ET is reading out on a weekly basis because we're measuring platelets and trying to normalize them.
Where are you doing the trials?
So the trials are essentially global. So they're running in the UK, in the EU, predominantly France, but also we're opening up in Spain and Italy, and then in the US. The ET study is not open in the US. We will go to the FDA in the next few weeks and open the protocol under the existing IND. So we've tried to encompass at least Europe and US geographies.
So you're moving fast into the clinic. Of course, nothing is free, especially when you're working in the clinic. You recently raised a little bit of money in a Series C financing, 38 million euros. Congratulations. Is this a proof point that investors are just sold on the potential of this approach?
I think it's a marker in the sand that clearly there's investor interest in the mechanism. I think we have been supported through 3 financing rounds since 2017 with investors that really believe in what we're doing. So we have investors that have supported us through all 3 rounds in BPI France, in Coma Partners as the founding investors, and Pontifax. And then we have Hadian and Sunstone that led the Series B, who also continue to support us. And we have VBio, who led the last round, and then all the others continue to support us. So I think we've got a growing investor base that understands the mechanism, that's really supportive. And that money gives us a runway through until the end of next year. So it will enable us to finish the phase 2 in ET. It will enable us to finish the phase 1 in lymphoma and the phase 1 in solid tumours, and therefore present us with quite a few choices in terms of what we're doing after that. We are now actively out talking to investors in order to put together another round as we can see phase 3 on the horizon. So we need to start thinking about how we prepare ourselves for a future phase 3 study in ET. So we will be looking to raise more over the course of the next 6 months. But yeah, I would say there's a growing level of enthusiasm for the mechanism, not just because of what we're doing, but because if you look in the literature, there are more and more papers coming out that describe CTPS1, the role that it plays in cancer, linking it to more biology, which again creates that sort of head of steam around a mechanism, because it's been difficult with us as we're trailblazing, first in class, with nobody else currently targeting the mechanism to therefore generate enough of a story around why this is interesting. And I should say that the reason that nobody else is working on this isn't because they haven't tried. It's actually because it's very difficult to find small molecule chemistry that's tractable to move forward with, that's outside of our intellectual property space. So we know other companies have expended quite a lot of time, money, effort, and not been successful in finding a way forward. So we continue to trailblaze, we continue to be first in class, but I'm sure that won't stay that way forever.
Does it help you when you're going out raising money, when you have this story around a pipeline and a product? So we can do so much. On the one hand, that's amazing. There's huge commercial potential somewhere down the line. On the other hand, does it ever come across as, well, we're going to be focusing on 3, 4, 5 completely different areas, and it's going to be that much more expensive to move through the process?
When you're early, I think it's really important to have more than one shot on goal. So I think in our early period of time fundraising, being able to demonstrate that we could do several different things, and therefore if one didn't work, we had another avenue, I think that has been really important. So I think at the last financing round, was particularly key that we were looking in several different directions. And at that time, we weren't even working on ET. So really the story was around solid tumours and that having a very broad opportunity, and lymphoma also coming through with positive data with signs of clinical activity in lymphoma patients. I think as we move on and we start thinking about phase 2 and phase 3, then I think we need to look very carefully in terms of what does the company need to become, and do we need to focus in, and rather than trying to do 3 different indications, does that become 2, because the solid tumours can also be multiple different indications. So I think there's definitely a balance to be played in terms of understanding what you can do, what the company can do, and in the end, what can be financed. And some of the later stage trials obviously need significant amount of cash, and therefore also a significant degree of focus when investors bring their money to bear on a phase 3 study.
So the money that you've raised right now, that's going all into the clinic, into these particular indications, leading you through to the end of next year?
Yes, so we've really made the conscious decision that everything goes into clinical development. We don't currently have a backup program that we're bringing through. Everything is resting on dencatistat. I think in the long run, there may be opportunities for us to bring other molecules into the company, other mechanisms into the company. But in terms of CTPS1 inhibition, it's all on dencatistat and it's all on these 3 clinical studies. But as I say, that will take us to meaningful readouts in terms of phase 2 and phase 1 proof of concept readouts for all 3. And then we can think about what the choices are in terms of where we go after that.
You're talking about a new fundraising round that you're preparing for now. You're out looking for money again. Have you considered as well partnering with Top Pharma? Have you considered going into some of this later clinical work with a partner in tow?
I think our challenge is we're a single asset company. So therefore forming partnerships around one indication versus another gets complicated because of the single asset nature of the company, so that we don't really have anything that we could license. So we can't license out for one indication and not the other, because it creates significant complexity in the end if we want to IPO or we want to M&A. So I think we've tried to stay away from that. I think there are opportunities to think creatively around China, around joint ventures to be able to move the program into China beyond what we're doing in the US and the EU. But other than that, I think it's more difficult for us. It's much easier if you've got multiple programs and then you can easily out-license one and keep the rest, and it doesn't cause too much of a disruption to the company.
Let's look a little bit forward now. If we're talking about the next key milestones for Step Pharma, what should we be looking for in the next 6 months or next 12 months? Firstly, I guess these first readouts from the trials.
Yes, so I would say in the next 6 months, 6 to 9 months, we will have phase 1 readout from lymphoma. And in the next 12 to 15 months, we'll end up with the phase 1 readout from the solid tumours in terms of the safety expansion cohorts. These are all open labels, so of course there are things that we can be sharing with people as we go along. And then also the ET study. So in the end, we'll get to the end of phase 2. Somewhere along the line over the course of the next 18 months, we should present some of that data at a medical congress. But clearly we're looking towards end of phase 2, end of next year, planning for phase 3. So those are the readouts that are going to come. There'll be other things that we're doing, like applying for orphan drug designation across the whole pipeline. So a number of different readouts that are coming. So there's a good news flow for the company over the next 12 to 18 months, essentially, in terms of what we have going on.
What about a little bit more broadly as well, just if we're talking about precision oncology as an approach within the industry? You know, these sorts of therapies that target the specific metabolic dependencies in cancer cells, how is that going to evolve? Is there still a lot to be discovered there? Are you still waiting on new science?
I think that the key there is they're fundamental pathways. So actually, if you're going to target metabolic pathways, you need to really understand how to do that in a very disease-specific way. Because the fundamental nature of the pathway says that in general, it should bring some toxicity with it if you block. So what we've identified is a way to target a metabolic pathway in a very disease-specific manner. I think you have other examples that also fit that bill. You've got the PRMT5 story with the MTAP deletion creating the synthetic lethality in a methionine salvage pathway. You've got the IDH1, IDH2 mutation that's giving you a 2HG metabolic synthetic lethality that you can go after. And so, that's a successful drug. The PRMT5s are coming through the clinic. There are others. There's asparagine and glutamine deficiencies or dependencies that are being exploited more preclinically, thinking about coming into the clinic. So I think there's lots of opportunity to target metabolic pathways. But you just have to figure out how you can pick out that disease-specific opportunity, which is often driven by that synthetic lethal observation that one element of a pathway is deleted, creating the opportunity to target the pathway in a different way elsewhere.
Andy, finally, if people want to learn a little bit more about the work that you're doing at Step Pharma, where can they go? Where can they find you?
So you can find us on our website, so at step-ph.com. There's plenty of information there on everything that's running, the clinical trials that are running, some of the background in terms of the biology. There's links there to all of the clinical trials on clinicaltrials.gov. And then, of course, you can find us on LinkedIn. We'll post things regularly on LinkedIn that relate to the work that we're doing, but that also relate to other things that might not warrant putting on the website, etc. So plenty of places where you can find us. So get online and search for us.
Fantastic. Andy, thanks for joining us today on Beyond Biotech.
No, it's my pleasure. Thank you very much, Dylan.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.